I changed my talk title a little bit today and decided to call it Taking Lemons and Making Lemonade, Clues from Graft-Versus-Host Disease to Design Successful Immunotherapy. And what I'm going to show you today is sort of a lot of the work we've done trying to identify the mechanisms that cause T cells to induce GvHD in our patients and what this can teach us for immunotherapy. So this is a figure from Bruce Blazer's recent review of GvHD in 2012, and it goes through all the steps of GvHD, and what we'll just go through it very quickly, and what you'll see is that a lot of the mechanistic steps that cause graft-versus-host disease are what we're actually trying to also induce for the leukemic antigen or the malignant antigen with immunotherapy. So you have your conditioning and your sort of the danger signals for the host. You get activation of T cells, both CD4 and CD8. Am I not loud enough? Usually loud enough. Okay. A cytokine storm that has some similarities to what we see with CAR-positive T cells. And then these T cells do their work, which is pathogenic, of course, in GvHD, but really something that we're trying to get to with immunotherapy, which is they identify their antigen, they seek out their antigen, they go into the tissue, they are able to tissue infiltrate, and then they destroy their antigen. And as I said, for GvHD, that's not a good thing, but there's a lot of, I think, important molecular parallels. And in fact, this is from a review that Sar Gillen and Carl June wrote last year, and if you look at each of the steps that they identify in a successful CAR T cell, they're actually very similar to what we see in GvHD. And I'm going to hit on some of these and what we know about GvHD and how they can help us understand immune therapy. And so while there's a lot that is known, really nicely depicted here by Bruce in GvHD, really black boxes persist at every step. And one of the things that my lab has been doing for the last over a decade now is trying to really decipher those black boxes. And what we have really tried to do is to take an unsupervised systems approach, so this is for you, Nick, to uncover mechanisms, try to really look at big data and see what we can figure out in terms of how we can better treat our patients with graft-versus-host disease. So we use a non-human primate model as well as mouse models and patient samples to do this. I think right now are the only lab in the country doing primate graft-versus-host disease, although there are a number of labs that we are working with now to sort of grow this throughout the country. So why do we use primates for this work? The immune system of primates really most closely resembles patients, and that's very important for a lot of mechanistic reasons, but really we're a very translational lab. We want to get new therapies to the clinic, and one of the really important things here is novel biological therapies are often cross-reacted with primate targets. And sort of as a value added over doing patient-only studies, we're able to do intensive multi-organ longitudinal analysis in primates, and I'll show you some work there, and I think it really helps us get some mechanistic insights. So several years ago now, we established the primate graft-versus-host disease model. We have a clinical model. I'll show you that. We understand more and more about the immunologic and genetic consequences of disease. And what we're doing now is using transcriptome analysis to identify new pathways of T cell activation and T cell infiltration. So here's the model in a nutshell. So we have recipient animals, we have donor animals. Just like we do in the clinic, we condition them. Mostly we condition them with irradiation, but we can use any type of chemotherapy. We do an apheresis using a clinical apheresis machine, the Optia Spectra, and then we follow them. And we can really dial in any kind of transplant that we want. So we can do an MHC match donor, we can do an MHC mismatch donor, we can do myeloablative, we can do reduced intensity conditioning, we can do standard of care GvHD prophylaxis, we can do targeted GvHD prophylaxis, and then, of course, one of the most important things we do is detailed immunologic analysis of immune reconstitution, function, bioactivation, and we now have some HIV studies where we're actually looking at HIV viral reservoir. But one of the big advances that we've made in the last several years is instead of to focus on mechanistic studies that are really candidate molecule studies, is to use transcriptome analysis to really take an unsupervised sort of data-driven view of the mechanisms that cause GvHD. And I'll show you some of that data just a second, but first I'll just show you sort of that the model works. So first of all, we can get good donor engraftment in our non-human primates. We use a GvHD clinical grading score that's exactly the same as what we use in patients, and we can see if you compare what we call our no RX cohort, so they get no immunoprophylaxis, versus substandard prophylaxis with rapamycin monotherapy, or sort of more standard of care methotrexate, we can see a graded response to immune suppression, and that reads out as a graded response in survival. And so we started out really with these 3 basic groups, but I think one of the strengths of the analysis that we've now been able to do is we've now expanded this, and we've transplanted many, many animals. And especially with respect to sort of when we're thinking about co-stimulation blockade or co-inhibitory blockade, one of the things my lab has been very interested in is co-stimulation pathways, and we've actually done some really, I think, interesting work, and I'll show you some of the data comparing CTLA-4 Ig, which targets both CD28 and CTLA-4, and directed CD28 blockade. So this has really become the data set by which we're really looking at mechanism now. So one of the things that we can do, in addition to just looking at the clinical scores, we can do some very detailed immunopatho or histopath, and I think this is going to be really important for looking at mechanisms of tissue infiltration. I think there's going to be some big crosstalk there with immune therapy. So we can look and quantify T-cell infiltration into the liver, the GI tract, the skin. Those are canonical organs of GvHD. We published last year in Blood that the brain is also an organ. This has been shown in mice now, and we've shown in primates, and there's increasing appreciation of that in our patients. And interestingly, this is a CD8 predominant effect, and I know we've had some discussion today. Why do all CAR T cells go to the brain, et cetera? It seems like with any inflammation, our T cells will go to the brain. And it has some sort of positive aspects and some negative aspects. So we don't only do systems-based approaches, we also use sort of multi-parameter flow cytometry, and through that we've really defined what we think are the canonical flow markers of GvHD in the primates, and we've gotten on now to show that they exist in patients as well. One of the first things we notice is there's a significant shift away from naive T cells towards memory T cell subsets during active GvHD. We see, as one might expect, a very highly proliferative phenotype, and something that I'll get back to because I think this is actually really also important for immune therapy, a pro-apoptotic phenotype of activated cells. Whether that's going to be exhaustion or death pathways, I think is really important. We have some data suggesting that it's probably both. In unprophylaxed GvHD, you get high cytotoxicity, and I'll show you transcriptome data that supports this as well. And we see evidence for both Th1 and Th17 phenotypes, and we actually think there's some mechanistic important differences between when you get one or the other, and again, I'll show you that. But as I said, one of the things that I became frustrated with about 5 years ago, 6 years ago, was that I was limited sort of by what I knew when I made my flow panel. So, if you only can test what you know when you do flow, and as you're trying to decipher mechanism, that becomes a real limitation. So I really wanted to sort of move into a data-driven approach, and we've done that with transcriptional analysis. And just to take you through our workflow here, so we have our large animal model. It can be also a mouse model or a patient model, canine, non-human primate. We do non-human primates. We purify T cells longitudinally post-transplant. And then for the studies I'm going to show you are all based on gene array because, like I said, we started these about 6 years ago. We're now moving into RNA-seq. For the first experiments, it was very important because nobody had ever discerned the primate transcriptome or any real transcriptome for GvHD before, is to do some internal validation both on clinical data and also on some protein expression data that we already had. And then we developed a pipeline for discovery. First, we wanted to test to make sure our transcriptome really looked like some of the other studies that had been done on GvHD. And then we got to the discovery where we're looking for novel targets of acute GvHD, and also this idea that we've developed, and I'll show you some data on what differentiates what would be called primary GvHD with breakthrough disease. And finally, to really validate this, we've developed what we call a multi-species, multi-assay pipeline, because if we're really trying to get these to the clinic, we want to have a very high bar of what potential molecules we're going to take through, because obviously the amount of work to take things to the clinic and the amount of risk to the patients is high. So in collaboration with Bruce Blazer, we run things through a small animal model, acute GvHD. We now have a very robust human MLR model where we can pick out molecules that really specifically are active in alloreactive cells. And then, of course, we're doing more and more patient samples. And if something hits all of these after going through our pipeline, we think it's a good candidate for clinical translation. So the first thing that we did when we looked at the transcriptome data was really look to see how well it mapped to our clinical data. And remember, here is where we can actually use all of our many cohorts now to look at how transcriptional changes really are read out in our clinical scores and histopathologic scores. So here we've taken the transcriptome that we got from our Affymatrix gene array, and we've done principal component analysis. And what you can see in principal component space, this is PC1 and PC2 here, that if you take those no RX animals, so they have severe GvHD, they are completely separated in transcriptional space from healthy controls, really indicating that there's something very different about these in their gene expression profiles. And then we can take each of our cohorts and map them onto this space. So remember that sirolimus monotherapy actually did very little to control GvHD. It just controlled it a little. And in fact, the sero group almost overlaps with the no-Rx group. As you march closer to sort of normality clinically, you also march closer down PC1 in transcriptional space. So TAC methotrexate is here. We've done a lot of work with CTLA-4 and I'll show you that as a monotherapy, CTLA-4 is not good, and when you combine it with other agents, it actually becomes very good, and you can see that in transcriptional space as well. So CTLA-4 as a monotherapy lies here, CTLA-4 plus sirolimus lies there. So we have a really nice indication that our transcriptome is going to mirror clinical outcomes, and then that, of course, gives us our a very important check to make sure that what we're going to see when we do the mechanistic studies is actually going to be predictive. And in fact, it looks very good. So if you actually just plot survival against PC1 for all the different groups that we've done, we get a nice correlation there. So really suggesting that this transcriptional work is going to hopefully be predictive of important mechanisms. So the first thing we did after we validated based on the clinical data is really looked at some protein expression data. Remember, I told you I spent years doing flow, and that looks really nice as well. So granzyme B is really a canonical marker of the primary GvHD of the no prophylaxis controls, as is Ki67. So these T cells become highly proliferative, highly cytotoxic, and we can see that this is true in our flow cytometry as well as in our transcriptome here. IL-18 was one of the canonical cytokines that we found to be secreted into the serum in patients, in recipients with GvHD, and we see that upregulated in our transcriptome. And as I said before, we see evidence of IL-17 both in our flow cytometry and in our transcriptional data. So not only could we do a little internal validation with what we knew from flow cytometry, what we could also do with this transcriptional data as it sort of cross-referenced with our clinical groups was really evaluate co-stimulation blockade or co-stimulation pathways for their impact on T cell activation. And again, I think this is one that may be interesting to this group in terms of why checkpoint blockade may be important. And so what we did is we tried to use our transcriptional analysis to understand the differential efficacy of two agents that we're very interested in, CTLA-4 and anti-CD28 co-stimulation blockade. So as many of you know, CTLA-4 is really the first co-stimulation blockade agent to make it to the clinic. There's actually two formulations that are FDA approved. Abatacept is approved for rheumatoid arthritis. Belatacept is approved for renal transplant. And they're very successful immunosuppressive agents. They've been around for decades. My lab has studied from decades. And really, they were the only agents available by which to block the CD28 CD80-86 axis until recently. There are now two different formulations of purely antagonistic, and of course this is very important with anti-CD8, purely antagonistic single-chain antibodies that are being developed both by Johnson & Johnson and Bristol-Myers Squibb. And our lab has had the pleasure of working with these over the last several years. And really being able to compare head-to-head these two pathways. And this is important because while an anti-CD20 antibody will be purely antagonistic to that CD28 CD80-86 pathway, CTLA-4 has the propensity to block CTLA-4. And so by comparing these, we can really try to dissect out what that impact is of additional blockade of CTLA-4. And this is what we found. So just as one might expect, if you looked at our canonical markers of either granzyme or Ki67, when you treated animals, and this is with what we would expect to be suboptimal blockade, either anti-CD28 as a monotherapy or CTLA-4 as a monotherapy. These are haploidentical transplants, T-cell replate. They get very bad disease. Actually, even with our flow markers, we could see that the anti-CD28 antibody inhibited both the granzyme B expression and the Ki67 better than the CTLA-4 Ig. Potentially predictable given the effect that CTLA-4 Ig is on CTLA-4 signaling. And in fact, that was translated into a survival advantage that was statistically significant. And without transcriptome analysis, we probably would have had to stop there. But instead, what we decided to do is sort T cells at that time where the animals treated with CTLA-4 Ig were getting sick, and those animals treated with anti-CD28 still looked great. And what we found is really interesting. So, and I think it really goes to the impact that targeting CTLA-4 may be on tumors. So what we find when we profile T cells, either that were treated with anti-CD28 and those animals are doing great, or CTLA-4 Ig, is 3 canonical changes happen. First, the animals that were treated with CTLA-4 Ig as monotherapy are really much more proliferative. So I've shown you just 2 gene sets that are proliferation gene sets, and we really see a nice signal for that, suggesting that when you also target that negative pathway, you get breakthrough proliferation. In addition, the T cells that come out of those animals are really skewed towards effector T cell genes. And so again, if you look at this GSEA plot of anti-CD28 versus CTLA-4 Ig, so here's the effector T cell genes that you see, effector gene list 1, effector gene list 2, you have a lot of enrichment for the CTLA-4 Ig, again saying that if you block that CTLA-4 negative pathway, not only do you get proliferation, but you also get increased effector function. And I don't have time today to tell you all about the leading edge genes, but they're really giving us some insights about what that CTLA-4 pathway may be doing. And finally, we see increased inflammatory gene expression in animals treated with CTLA-4 Ig again. So both an inflammatory gene list and also an antigen response gene list. So I've just given you a taste of what we can do with this, and really what we're doing now is interrogating those gene lists to find what we think will be CTLA-4 specific molecules that are responsible for the better outcome when you don't also block this inhibitory pathway. So in addition to doing some fun work with the CTLA-4 Ig and anti-CD8 work, one of the other things that was very important when we did this study was to really interrogate what we know are pathogenic drivers of GvHD and to see how they related to our different treatment cohorts. And to do this, it was very important to use a comparison not of the no-Rx cohort to healthy controls, because remember that during transplantation, not only do you have alloreactivity, but you have homeostatic proliferation. So it was very important to take that homeostatic T-cell activation out of the picture. And we did that by having an autologous transplant control. So all of the next comparisons I'll show you will be between animals that were undergoing an alloreaction versus animals that were post-transplant but had no alloreaction. And what we see is really interesting. So it's very clear that when you have animals that have no prophylaxis, that are really just going into GvHD with no control whatsoever, they really have a skewing towards that Th1 profile. And I think that the individual genes may give us markers of key activation markers that we may want to target positively during immune therapy. So really all the canonical markers are there. This is CCR5, CD2, IL-12 is very important. But as many people have noted in their talks, not only are these activations sort of canonical activation markers, but we also see upregulation of this gene HAVCR2, and it's important to know that's TIM3. So we have upregulation of TIM3, we have upregulation of LAG3, we have upregulation of PD1, and so there really, we think, is a canonical exhaustion phenotype that comes with activation. I think that these studies may give us clues as to the less appreciated ones that may be important. So we think that there may be exhaustion accompanying Th1 skewing, but what we also found that was fascinating to me was a really strong cell death signature. So all of these cells, and we know that when T cells get activated, they die, and this is something that we really want to block with immune therapy. And what we were able to show is in these untreated animals, they had multiple death pathways that were upregulated, and the markers that we're seeing are really interesting, things that I had actually never thought of before, but not only necroptosis, apoptosis, but also pyroptosis, and the inflammasome seems like it's going to be very important in probably the natural downregulation of T cell responses so that you don't become a big lymphoma, let's say, when you've got GvHD or any other T cell response. And I think these actually may identify novel targets that could improve CAR T cell survival. So we're really interrogating these very closely now, too. So one of the things that we're able to do is not only to validate internally and identify which pathways are involved, but really use this as a pipeline for novel, to understand novel ways that T cells get activated with chronic antigen. And one of the things that just comes up over and over again in these arrays, you can see it from across the room, is that cyclins and kinases are all over the place. We see so much upregulation of multiple kinases. Here are just a couple of GSEA showing CCNA2 pathways, CDC2, CCNB1, BUB1 is a huge cyclin that is upregulated in our arrays. And one of the ones that's come up is to be very potentially clinically relevant is Aurora kinase A. So no one had ever really looked at it in GvHD before, but what we found is it was very highly enriched in our NoRx and our sirolimus monotherapy compared to autologous controls. And not only that, when we put it through this pipeline, in a mouse model, it prolonged GvHD-free survival. When we looked at it in allo-specific MLR assays, so we took the allo-proliferating cells and compared those to non-proliferated cells, it was super upregulated in both CD4 and CD8 genes. And finally, really importantly, when we now are developing a big biorepository of purified T cells from patient populations, it's upregulated in those too. And in fact, all these assays together make it our first candidate that really has made it successfully through all the different stages of our pipeline, and we're now in the process of trying to get this into the clinic for patients with GvHD. So we've talked a lot about the different pathways that are upregulated. One of the things that really just confuses me, right, and I think all of us who treat patients with GvHD, why do some patients get only skin? Why do some patients get only liver? Why do some patients get only GI? What targets those T cells, and how can we specifically block that T cell infiltration? Or for immunotherapy, how can you specifically drive a T cell to a different tissue? And we think we can use some transcriptional analysis of our primate model to look at that as well. So again, going back to the June review, remember that one of the most important issues that we have, of course, especially with solid tumors, is trafficking to the specific site. And what we've been able to use is our primate model. Here again, we're going back to the anti-CD28 to really start looking at that. So it's a little hard to see here, but this is our anti-CD28 monotherapy and then anti-CD28 Rapamycin. So the monotherapy does well, but eventually these animals get GvHD. The anti-CD28 Rapamycin has been kind of a home run, and we actually think it's going to be a really important treatment strategy or prevention strategy for GvHD going into the clinic, and we're working on a trial right now for that as well. But both of these had T cells infiltrate the liver. And you can see there's more T cells from the anti-CD28, but there were some in the anti-CD28 Rapa animals. And so we were able to not only get transcriptomes from the blood, but also from the organs in primates, and it's starting to allow us to get some insights into the mechanisms that drive T cells into tissues. And so the first thing we did was did the comparison of T cells in the liver and in the blood to see what was different about these T cells. And what we find is that for both FR104 anti-CD28 alone and the anti-CD28 plus rapamycin, if you look at those T cells and compare them what's in the blood, they're effector cells. It's kind of a no-duh situation here, but what we were able to do, again, by really interrogating closely the leading edge genes and asking specifically how they differ for different organs, we think we're going to be able to identify the really important drivers into those tissues on a organ-specific manner. But maybe even more important than that is to ask this question of, so there are T cells in the liver in the FR104 Rapa, but they're not doing anything, right? So they're getting there, and T cells do get into organs, but they're not doing anything. So this allowed us to say, well, what differentiates the T cells that get in the liver and cause disease in anti-CD28 alone, and those that get in there just because they got in there in the FR104 Rapa? So we did that comparison too, and the results are really interesting. So what we found, first of all, is there's some really important chemokines that are getting upregulated, CCL1, CXCL13, but really what was surprising is the IL-17 pathway starts showing up here. So POU2AF1 is a gene that actually Alex Shalick identified a few years ago in a TH17 subset, as well as IL-21. And it started us down the road of thinking that IL-17 and the shift towards an IL-17-based mechanism may actually define T cells that break through immune suppression and get to the tissues. So we've really started going down this road hard, and the way we've done it, again, is to make use of the fact that we've now done many, many transplants with many, many different immune modulation strategies. And what we've found is that we can actually group our transplants into three, or our T cells that we pull out of those animals into three groups. We have one group that we call primary, and these are animals that either have no prophylaxis or have such bad prophylaxis that they get horrible GvHD really quickly. And then we have the breakthrough GvHD. So at one point, these animals are suppressed. They actually are doing fine. They don't have clinical GvHD. And then eventually they break through. And when you see, eventually these breakthrough animals die of their disease, but their survival is much better than the primary. And so this has allowed us to now use these bigger classifications to really gain statistical power and try to understand what the mechanistic drivers of these differences are. So first of all, you can identify the different groups with transcriptional analysis. And again, here's PCA space showing that if I look at PC2 and PC6, I can really pull apart those three cohorts, which is really important. So if you look at the primary, you'll see that it's just like what I showed you for the NoRx, is that it's a very Th1 skewed cohort where you have upregulation of a lot of canonical Th1 drivers, IL-12, CCR5, CXCR3. And in fact, if you look functionally, T cells in primary GvHD animals elaborate interferon gamma, but not IL-17. So we know what primary looks like, but we really wanted to delve deeply and specifically into what we call breakthrough. To do that, we took those transcriptomes from those breakthrough animals and tried to compare them to every other group we could to really get a very breakthrough specific gene list. So we compared them to autologous controls, we compared them to healthy controls, we compared them to primary, and we took the breakthrough specific genes of all those comparisons and ran analyses on them. And what we find here is really interesting because very unlike the primary, here you see upregulation of many of the canonical markers of Th17 cells, so IL-17A, IL-22, ROR gamma C, and downregulation of those canonical Th1 cells. And we think this is really important. These are cells that they're chronically exposed to antigen. What happens in that setting of that chronic exposure? They move away from their very active state, not necessarily to die, but to take on this Th17 phenotype. And in fact, when you look functionally, not only do they look like that transcriptionally, but the cells make IL-17 an important control. And so now what we're doing is not just looking at those individual genes, but using network analysis to ask, well, what are the correlations between those genes? And we're just starting this analysis, but we've already gotten some really interesting results, and here's one of them. So if you look at the correlation module right here, and you take out just, in fact, a random, highly correlated gene, what we come up with is a gene for proliferation, MCM3. And remember, T cells usually, when they proliferate, start to die, right? And so sometimes you don't have to worry about them pathogenically, or for immunotherapy, they go away and you don't want them to go away. Well, in these animals that just have persistent T cell activation, still have pathology, what they do very unusually is they upregulate survivin, right? So it really implicates the upregulation of anti-apoptosis genes as potentially a pathogenic driver in GvHD, but also will start to identify genes that you may want to upregulate or enforce the expression of to try to enforce survival of the T cells that we put into our patients with malignancy. And so all this work has really led to a model that we're looking at both, again, in primates, but also now in patients, where in the post-transplant period, when you have the introduction of antigen that's there all the time, remember this is a really good model of chronic antigen, what happens first is robust proliferation is skewing towards Th1 profile if you don't have good immunosuppression. But if you have pretty good immunosuppression, this is what we see in our patients after transplant, you actually really nicely dampen that Th1 skewing. But what the escape mechanism for those T cells is really a Th1, Th22 skewing, and we think that's responsible for the ultimate disease that these patients have. So in conclusion, what I've shown you is that, first of all, there are many parallels that exist between T-cell activation during immunotherapy and T-cell activation and tissue targeting in GvHD. And the primate GvHD model has allowed us to interrogate the mechanistic drivers of T-cell programming during GvHD. We think it's provided new insights into the therapeutic advantage of specifically targeting CD28 versus targeting CD28 and CTLA4 with CTLA4 Ig. And for the immunotherapist, it's really the subtraction of that gives you some of the mechanistic drivers of blocking CTLA4. It's allowed us to identify multiple novel kinases involved in T-cell antigen-driven proliferation, including Aurora kinase A, which we think is really important, and has identified a shift from Th1 to Th17 mechanisms during T-cell activation that occurs despite immunosuppression. So in conclusion, we think these studies provide new candidate mechanisms and molecules that are critical for persistent T-cell activation and tissue infiltration in the presence of persistent antigen. And these might be amenable for testing as engineering targets for T-cell immunotherapy. So with that, I will stop and I'll thank my lab. I want to give a big shout out to the 3 postdocs who have done a huge amount of work to get all this together: Ben Watkins, Victor Tkachev, and Scott Furlan, who are all here today. So thank you very much.